Gas chromatography is the most popular organic analytical technique in the world. Virtually all major companies have GCs available for research and quality control. The fundamental limitation of GC is sample volatility. GC samples must be able to be vaporized within the temperature limits of the GC heated zones, typically 400 degrees centigrade or less. Completion of this introductory training module on gas chromatography will prepare you to describe gas chromatography and a gas chromatograph, identify the main components of a GC system, list common industries and applications where GC is used, select samples that would be analyzed by GC, identify capillary and packed GC columns, describe the role of the column in GC separation. Explain the terms sensitivity and selectivity as applied to GC detectors. List various ways that samples are introduced into a GC. The gas chromatograph is used to analyze samples from many different industries for quality control and for research. GCs are used for the development of new products from the petrochemical to the pharmaceutical industry. The GC is also used in environmental and clinical labs to determine levels of toxic chemicals in the human body, in our agricultural products, the air we breathe, and the water we drink. Shown here are some typical products that could be analyzed by a gas chromatograph. GC samples have molecular weights from 2 through about 800 or slightly higher. The GC sample must be volatile, that is, able to be vaporized without unwanted or unexpected thermal breakdown, at temperatures below 400 degrees centigrade and swept through the column by the carrier gas flow. GC sample concentrations are in the percentage range down to parts per trillion. Here are some other types of GC samples. Most commonly, liquids are analyzed by injecting them into a heated GC inlet or injection port using a 5 or 10 microliter syringe. Gases can be analyzed using a gas-tight syringe or a gas sampling valve. Solids can be analyzed by extracting chemicals that are in the solid with a liquid solvent. Think of making coffee. Or by placing the sample into a sealed vial and heating it up. As the sample is heated, volatile chemicals will be evaporated into the headspace of the sealed vial, which can then be injected as a gas into the GC. Think of how you smell chemicals in a new car that has the windows closed and is sitting in the sun. The car has become a huge headspace vial, and your nose is the detector. When injecting liquids, the speed at which the liquid enters the heated GC inlet is important. The liquid must be injected out of the syringe before it is heated and evaporates from the syringe needle. Otherwise, variable amounts of sample will make it onto the column. The need to inject quickly into the hot inlet has led to the importance and ubiquity of autosamplers. Using an autosampler allows for up to 150 samples to be analyzed automatically and to ensure that exactly the same amount of sample gets injected every time. There are 5 primary components to a GC system. Carrier gas, from a pressurized cylinder or a gas generator. Typical carrier gases are helium, hydrogen, with safety precautions, and nitrogen or argon. The gas must be very pure, greater than 99.9995%. The carrier gas carries the sample through a GC column, where it is separated into individual components of the sample mixture. Sample introduction. The most common method is to inject 0.2 to 3 microliters of liquid into a heated injection port using a syringe. The requirement for GC samples is that they must be able to be vaporized in order to be carried through the GC column. Column. The column is the heart of the GC. It contains a very viscous liquid called a stationary phase that is used to separate the sample by virtue of solubility and boiling point. The column is temperature controlled in an oven. Detector. After the sample is separated, it exits the column into one of many different types of detector where the amount of sample present in the original sample mixture is measured. A computer chem station or integrator is used to measure the amount of sample components as they pass through the detector, and also to identify those components based on retention time. The column is the heart of the gas chromatograph, where the sample components are separated. Shown here are examples of capillary and packed columns installed in various GC ovens. The entire GC analysis cycle can be divided up into 5 steps. The first step deals with sample preparation, which is often the most time-consuming step, depending on the complexity of the sample matrix. An example of this step is extracting pesticides from fruit or soil. The injection step involves rapid vaporization of a liquid sample with subsequent mixing with the carrier gas and introduction into the GC column. The analytes are then separated in the GC column with a variety of physical and chemical separation mechanisms. After the separation of the analytes, they are individually measured in a GC detector that converts some chemical or physical property into an electrical signal. A PC-based data system, like the Open Lab chemical data system, is used to process the digitized signal from the GC and use various mathematical calculations to determine peak areas, peak heights, peak widths, and retention times. The following will depict the basic components of a GC system and also show the process of sample injection, separation, and detection. This shows the cross-sectional view of a capillary GC column. The column is made of fused silica glass that is coated on the outside with polyamide, a high-temperature polymer that allows the column to be coiled and bent without breaking. The stationary phase is typically a highly viscous liquid like methyl silicone that is chemically bonded to the inside surface of the glass column with a thickness in the range of 0.1 to 3 microns. The syringe with the GC sample to be evaluated is inserted into the heated injection port through the septum. The septum is a rubber seal that isolates the GC carrier system from the outside atmosphere. The plunger is depressed, injecting the sample into the hot injection port. The injection port is sometimes referred to as an inlet. The inlet usually has a glass liner or sleeve that provides an inert area for the vaporization of the sample. There are different designs and internal volume liners depending on the application. When the sample goes into the heated inlet, it is very rapidly vaporized. This results in a momentary increase in the pressure inside the inlet. Once the sample is vaporized, it can then be swept into and through the column by the carrier gas. The sample moves in a tight band into the GC column, which is initially held at a low temperature. The lighter, lower boiling point solvent moves away from the sample components that are being analyzed. towards the detector. The first peak in a GC analysis is usually the solvent peak. It will be very large and often saturate the response of the detector. As the GC oven is temperature programmed, the carrier gas starts to sweep the sample components through the column. The time that it takes for each sample component to reach the detector will be a function of its boiling point and its solubility in the stationary phase. Notice that the column oven temperature is being increased by a predetermined and accurately controlled temperature program. The oven temperature is programmed up and the separation continues. The first component reaches the detector and a peak is displayed on the integrator or chem station. The height of the peak or the area under the peak will be used to determine the amount of each component in the original mixture. As the temperature program continues, each peak elutes from the column and is detected. The chromatogram, which is the graphical representation of the sample elution and response, is complete. Each peak is identified by the specific time that it leaves the column and passes through the detector. This is called the retention time. Each type of detector responds differently to compounds. Non-selective detectors will detect anything. The FID detector, for example, will detect anything organic. Selective detectors are designed to detect only specific compounds for analysis. The NPD detector, on the other hand, will detect nitrogen phosphorus only. Selectivity is the response of one group of compounds in a detector relative to the response of another group of compounds in the same detector. Sensitivity is technically what change in response is achieved per a change in sample concentration. The MDL speaks to how low of a concentration a detector can still provide a measurable response for when a measurable response is defined as a peak height 2 to 3 times the noise level. This is a graph that depicts the detection limit and the dynamic range for each of the more common GC detectors. GC peak times really do tell the whole story of the composition of the sample being analyzed. A typical chromatogram is shown. Retention times are used to identify the components in a mixture by comparing sample peak times to known reference standard peak times. If the times match, it is very likely that the components are the same. The retention time is determined at the apex of the peak. Peak height or area is used to determine the amount of components present. Standards of known amounts of the components can be used as a reference in a calibration procedure to produce quantitative results that report the amount or concentration of each calibrated peak.